When emergency first responders were overwhelmed by Los Angeles County's most destructive fire yet, a band of surfers, along with their neighbors and friends, stepped up to defend their home turf in Malibu. Their devotion to home drove them to show up for their community during the fire and for years afterward. And now, a model they call the Community Brigade program could change everything, leading to more lives and more homes saved during the increasing wildfires across not just California, but the world. Join reporter Adriana Cargill from KCRW, NPR's All Things Considered, Crooked Media, and more, as she investigates a wildfire story that is not depressing, but is, in fact, a clear hope for the future. Listen now to Sandcastles, an award-winning podcast about home, how we create it, and why we fight so hard for it. Hi, everyone. It's Quinn. We are back on our Good News series today, and I am delighted to offer up Can Zebrafish Beat Childhood Cancer? With doctors Jaclyn, Taroni, and Genevieve, Kendall on as our guests. They are cancer scientists working together with one of our favorite charities, Alex's Lemonade Stand. We had so much fun talking to these incredible women. Shocking that they even had a conversation with us. But we learned a ton too, and I think you will too if you haven't heard this one before. And honestly, who in their right mind doesn't want to beat kids' cancer right back to where it came from, you know? So please enjoy this one. They're making so much progress in these amazing places. They're using super cool new data to do so. And of course, Zebrafish, which I highly recommend you Google image. It's the coolest thing ever. Anyways, I hope you enjoy this one as much as we did. Thanks as always for listening. Welcome to Important, Not Important. My name is Quinn Emmett.
And my name is Brian Colbert Kennedy.
This is the podcast where we dive into a specific topic or question affecting everyone on the planet, this planet right now, or in the next 10 years or so. If it can kill all of us or turn us into something super cool and futuristic, like, I guess, controversially, Starship Troopers, we are in. Our guests are scientists, doctors, engineers, politicians, astronauts, even a reverend, and we work together towards action steps our listeners can take with their voice, their vote, and their dollar.
Why are Starship Troopers controversial?
It's the whole thing. That's a fun talk question, Brian.
Got it. Hey, this is your friendly reminder that you can send questions, thoughts, feedback, and more to us on Twitter at importantnotimp or email us at funtalk@importantnotimportant.com. You can also join thousands of other smart people and subscribe to our free weekly newsletter at importantnotimportant.com.
You can also leave us a voice message at the link in your show notes. Those are super fun, and we'll include you in the show. Most of them are too weird that we get. And we get plenty of them, but appropriate ones, which honestly is saying something if you've listened to Fun Talk. Appropriate ones, we will include those.
Will we? Because we haven't yet.
That's what I'm saying. They're all too weird, which that's what I mean. Like the bar is low. Anyways, hey, this week, Brian, we're getting back into childhood cancer because fuck that, man. And this week we're getting technical. We're talking really nerdy about beating childhood cancer with some very unusual and exciting and tiny and glittery, at least to us, resources.
Yes, we are. Very exciting, very scientific-y talk, but it was very cool.
Why don't you tell them who stopped by Mr. Brian's neighborhood today?
Oh, sure. Our guests are doctors Jaclyn, Taroni, and Genevieve, Kendall.
They came on the podcast after listening to previous ones, which is always a good test. And they work with probably our favorite place on the planet, Alex's Lemonade Stand, to just beat the shit out of childhood cancer. And I admire and respect, and I'm so thankful for these ladies.
And they were really fun to talk to. It was a good conversation.
It was really good. So let's not keep it going. We're already at 3 minutes for this intro. Jesus.
Oh.
All right, let's go talk to doctors Jaclyn, Taroni, and Genevieve, Kendall. Our guests today are Dr. Jaclyn, Taroni and Dr. Genevieve, Kendall. And together we're going to ask, Brian, can zebrafish cure childhood cancer? Dr. Taroni and Dr. Kendall, welcome.
Thanks for having us. Yeah, excited to be here.
We'll see.
I think you're going to be happy all the way up until we say goodbye. All right. Honestly, thank you very much. And if we could just get started by each of you just sort of letting us know who you are and what you do.
So I'm Genevieve Kendall, and I'm a postdoctoral scholar at UT Southwestern Medical Center. And I study a pediatric muscle cancer. And I utilize zebrafish models in order to understand how these cancers develop and then implement these models to better identify new therapies that we can use in the clinic.
That's pretty awesome. Jaclyn, go ahead, try to match that.
Ooh, tough. I am Jaclyn Taroni. I am a principal data scientist at the Childhood Cancer Data Lab, which is an initiative of Alex's Lemonade Stand Foundation. And my job is to understand how folks like Genevieve and other childhood cancer researchers can use data to answer the really important questions in the childhood cancer field.
Awesome. I love it. This is one of those some of our conversations are more ethical. Some of them are more morality-based. Some of them are topics that people generally know about, like why is the ocean cold? And then sometimes there's really technical ones like this. And I like to imagine like where, if someone said, Brian and Quinn, figure out how zebrafish relate to cancer, like where we would start. Literally, like what is step one? And I cannot even imagine how that would begin. So this is going to be educational
pretty
for
excited.
all of us.
All right, so Quinn is going to go over some context that I don't know where he's got this information, but he'll talk to you, guy on the street, and then you can, of course, correct him please correct him as much as possible. It makes me very happy. And then after some context for our question today, we'll get into some action-oriented questions that get to the heart of why we should all care about it and what we can all do to help support you ladies. Sound good?
Sounds good.
Sounds great.
To be clear, like listeners, we've talked about this before. It's kids' cancer. Like if you don't care about it, you're a monster. It's just it should be pretty obvious. Anyways, listen, before we get into that, both of you, we do like to start with one important question to set the tone here. Instead of saying, tell us your entire life story, if you could each answer, why are you vital to the survival of the species? That would be great.
Okay. No, that's a great question. So I was thinking about this question because I've obviously listened to your previous podcast, and that really, to me, I do what I do because I'm passionate about it, and I'm trying to make this world a better place and to help kids. And I think even more importantly than that, I'm really dedicated to training the next generation of people that will take up this task and continue this work and continue to, spread knowledge and, give back. And so I think that's really what we need.
I love that. Pay it forward. Pass it on.
I would say I'm not. I think folks like Genevieve, who are real childhood cancer experts out there training folks, I would say are probably more important than I am. But I'll echo a lot of what Genevieve said, which is, I think as scientists, we want to be in a spot where the folks who come behind us have better resources, tools, training to answer those really important questions. And I think in my position, that's really important to me and something I think about a lot.
Jackie really elevates what I'm able to do, though. And she's incredibly thoughtful in helping design experiments, cull big data, and make the biggest impact that we can make for trying to identify cures for childhood cancer. So I think you're being humble, Jackie.
Oh, thank you.
Awesome. I love it. So on that note, both of your life work, which sounds like it's fairly complementary to each other, which is awesome, involves doing immense and just incredibly impactful good for a group of young, fragile, just getting started humans who they haven't been smoking their whole life. They didn't deserve these illnesses that they have. You are truly giving yourself to something greater than you. And thankfully, so many of our conversations are with folks like the both of you. There is a lot of good in the world, and we try to expose that and give a voice to as much as we can. For each of you, is there a specific relationship that you can point to that was a catalyst to get you to where you are today, to do what you do?
So I had a really influential biology teacher in high school. And ever since I took this class in 9th grade, I knew I wanted to be a scientist. And it was a bit of a road to get to pediatric disease. I started doing research trying to understand how South American bats were related to each other at the genetic level. And after that, I realized I wanted to help kids. And so ever since then, I've been trying to understand, how we can identify new therapies for pediatric muscle disease. And so it's been incredibly rewarding because I do get to interact with these kids that have cancer in their families through Alex's lemonade stand and through being next to a children's hospital and try and show them what I'm doing and try to make the biggest impact possible. And so that's really rewarding and motivates me to even work harder.
My 9th grade biology teacher was Mrs. Thompson, and bless her heart, she did not make you want to care about science.
Oh no.
That's too bad.
You could have been on a different path then.
Yeah, I think so.
You could have been studying bats.
Right. Yes, exactly.
Right.
That's awesome. That's really wonderful.
So for me, I think I got the research bug probably in college. I had a wonderful professor named Dr. Bean.
Amazing.
Who took me in when I was a junior in college, and I really got the bug. I feel like my upper level biology courses, I was always so struck by what we didn't know. And that led me to go to graduate school. And interestingly enough, I thought I would be like an immunologist. And when we go to graduate school in the biomedical sciences, we often do these rotations where we spend like a 2 to 3 month stint in different laboratories. And the one that I started off in was more computationally focused. And I really liked that. And I landed in a lab that worked on a rare systemic autoimmune disease. And a lot of that work was basically asking the question, which is about, if I'm working on a rare disease and I want to use techniques like big data that, require a lot of cases, how can I leverage other types of data or data measuring things other than what I'm directly studying to learn about the disease that I'm interested in? And that is how I see a lot of what we do at the Childhood Cancer Data Lab as well, is thinking about all this investment that's been made in different kinds of contexts, looking at cell lines, looking at zebrafish. How does that come together to teach us about childhood cancer? And that is very exciting to me, to put that data to work.
Do you think the same opportunities to do that, and we'll get more into this, would have been available 10 years ago before these advances in algorithms and data compilation and organization?
I don't think so. So the kind of data we work on, it's called gene expression data or transcriptomic data. Luckily, there's always been a very strong culture of data sharing in that particular realm. But if you look at kind of the expansion, the number of assays that become publicly available over time, as sequencing a particular kind of technology became more ubiquitous, you kind of see it take off like a rocket in like 2008. So it's relatively young in the scheme of things, this explosion of available assays.
That's awesome. Well, I'm glad you're right place at the right time. Obviously, a whole lot of people are benefiting from that. So again, just some quick context here. Let me blow through this as mostly catching people up who maybe listened to our previous episode with Jay or did not. So we first dug into pediatric cancer back in episode 48, titled "Why the Hell Does Kids Cancer Even Exist and What Can We Do About It?" And that was with Jay Scott, now a good buddy of mine and the co-executive director of the Alex's Lemonade Stand Foundation, which was started with her very own little lemonade stand by his late daughter, Alex. If you have not listened to that conversation, it is fairly timeless, as our best episodes are, and I promise it will hopefully inspire you and probably make tears explode from your face, as it did mine, all over my keyboard. But during that episode, we focused a lot on and just so you guys understand, we'll have 12 different conversations about the ocean, for example, from did Fukushima dump radioactivity into it to, how are the jet streams changing the ocean to funding to food stocks off the coast, so that people really get a fuller context of what's going on, because all these things are connected. And when it comes to pediatric cancer, in that episode, we talked a lot about sort of the macros. We talked about how cancer is the number one cause of death by disease for kids under 19. Yet, despite that, childhood cancer research receives just 4% of funding from the National Cancer Institute. And for reference, cancer overall is responsible from what I gather, about 29% of American deaths. We discussed how childhood cancer is not just one disease. There's about 12 major types and over 100 subtypes. Like anything with cancer, when people say we're going to find the cure for cancer, that's not how it works. Shit's very complicated. And we talked about how there are some cancers kids are more likely to get and the progress we've made against them in research, and treatments, and recovery, and living with it. We talked about Alex and her life and her diagnosis and her treatment and her passing and what all of that meant for Jay and his amazing wife, Liz, and how that changed things for them, obviously in a number of ways. And of course, we talked about the enormous impact that Alex's lemonade stand has had in the fight against pediatric cancers. I'm a big supporter in a number of ways. I know Brian is, and listeners, you should be too. But today we're going to get into some nitty-gritty science. It's going to be fun because Alex has started something new recently, and we've talked about a little bit, called the Childhood Cancer Data Lab, or CCDL if you're in the know. And it is already revolutionizing the way that childhood cancers are researched. So on that note, let's dig in a little bit to our completely clickbait title of "Can Zebrafish Cure Childhood Cancer?"
Yeah, right.
Jaclyn, can you kind of finish getting us up to speed? I guess tell me what CCDL is and why it exists.
Sure. So the CCDL was started in August of 2017, and we are a team of user experience designers, engineers, and scientists. And our goal, in essence, is to put big data in the hands of childhood cancer researchers who are poised to make the next big discovery. So there are several parts of that. Essentially, there's barriers sometimes to using these data to make discoveries. That can be in access. It can be in knowledge. So part of our mission is software, building tools to make these data easy to use. Part of that is training. So Genevieve is one of our first training participants, and we're very grateful to have her come out and give us feedback on that. And part of it is scientific workflows. So bringing things in from different fields, using our expertise, and putting that to work for childhood cancer research.
That's awesome. Where did, and I feel like I've asked Jay this question, and I've either he avoided it, as he does with most of my text messages, or I've forgotten.
He does too. My God, I do the same thing to you.
This is not that conversation, Sorry. Brian. We're not doing this right now. How did CCDL come about? Where did was there another similar model and another vertical that inspired it? Was it just clear that something like this needed to exist for you guys?
Yeah, so I have to definitely give props to Jay and Liz for, Jay describes it as just in their gut. They felt like this was a missing piece in the field. But this is definitely kind of first of its kind stuff. There's no other informatics lab run by a funder devoted to childhood cancer, as far as I know.
That is super rad. That is very cool.
So one of the issues that CCDL is addressing is with all of these new sequencing technologies, everybody's potentially using a different platform, a different pipeline, and then they're making the data publicly accessible, but it's all in different formats. And then for people like me, it's difficult to harmonize that kind of data. And so the CCDL provides this expertise that I can draw on in order to identify data that's important to me and integrate it and then leverage it for my own work.
Got it. That's very helpful.
Seems crazy that it didn't exist before,
It's just a lot of people working in silos, essentially, and doing whatever experimental protocols, work best for them in their hands. And there's no standardized technique, I would say. Jackie can speak more on that than I can, really. But
so that's definitely true. It has a bit to do, I think the CCDL model is powerful because it's happening outside of, to a certain extent, the way data is typically generated. And what I mean by that is it's an independent party run by a funder. So I am not an assistant professor. I'm not in academia per se. And I think what we're interested in is we have the room to really focus on the cause in ways that some folks in academia don't always have the same opportunities because our incentive structure is a little different, if that makes sense.
Yeah, I think that makes sense. Yeah.
So we have the space to focus on what doesn't work best for us, our particular experiment. We have the space to kind of look more broadly and bring it all together because that is how we are judged as successful.
Sure, that makes sense.
I don't want to rag on academics. I don't mean it like that at all.
I think most people know that like academia is a incredible pursuit and an invaluable resource and a just labyrinth nightmare of red tape and things like that. Not even including a case like this. Just getting into tenure or anything like that. It's crazy out there,
and it's really hard. Also, it's hard to do what we do. Requires a lot of resource and a lot of expertise in things like software engineering that folks don't always have the access to resources to do that stuff.
Right. The CCDL is a really unique model. I don't know of any other similar model where they do have the resources and the expertise in the same place to tackle this problem.
It's almost like the old Bell Labs example.
Exactly.
Where it was a bunch of white guys in that period, which they were great, but I'm glad we're moving beyond that. But, having those resources in the same building makes a big difference. And obviously it's 2019, so we don't need to be in the same building anymore. But I imagine in academia, you probably have those resources in the next building, but the effort to integrate with them in any way is probably just infinitely more complicated. For a number of reasons, but also just like you said, you guys are judged and measured differently, very differently.
That's true.
Yeah. Dr. Kendall, when we first got connected, you responded that you would like to discuss, quote, Genevieve's use of zebrafish genetic models to study pediatric sarcomas and the high dimensional assays we focus on in the CCDL as part of her experimental toolkit. So let's get into that, like to the most very basic level, like I am a child, peanut butter and jelly, if you know what I'm talking about.
Sure. So pediatric cancer is usually, genetically based, and there are very few mutations in genes that cause these cancers. And so the specific cancer that I study is a cancer of the muscle, and it's called rhabdomyosarcoma. And it's caused when two genes fuse together and form this like aberrant gain of function protein that acts in the cell to turn off and on hundreds, if not thousands of genes inappropriately. And so we've identified this gene as the cause of this muscle cancer now 20 years ago. But we have no drugs that target it directly. And so this is really frustrating to me and how I got into this field. And so what I do is I take the human form of the cancer gene and I actually use genetics to integrate it into the zebrafish genome.
So just to dial that way back for both me and, as I like to blame them, our listeners who are texting and driving down the 405 right now, why the zebrafish? I need a little more context on that. Trying to understand. The things that are going through my head are probably very different from how you're about to describe them.
No, so zebrafish, although they look very different at the genetic level, they're very similar to us. So over 70% of their genome is conserved with the human genome or is similar to it. And over 80% of human disease-causing genes, a similar zebrafish gene. So they're very amenable to understanding human disease and these, developmental pathways that take us from a single cell all the way up to an entire organism are very similar. And so they have a lot of experimental advantages that you don't find in other model systems. And one of them is that you can use them for like large scale drug studies. And so baby fish can develop in 1 day, which is really fast. And you can actually put hundreds of fish like in a plastic dish that's the size of your hand and screen 100 compounds at a time for activity in the fish. And I literally got into fish because I was sitting in the audience at a muscle conference, and these fish like couldn't move because it had a mutation in its muscles, and they added a drug to the water and the fish could suddenly move again. And after that, I thought, I absolutely need to learn how to work in this system. And that's how I got into it in the first place. And I was shocked to learn that when we insert these human cancer genes into the zebrafish genome, they actually get cancer that looks exactly like the human disease. When you look at the structure of the tumor cells themselves, or when you look transcriptionally or at all of the gene expression changes, which is what I do in collaboration with like Jaclyn and the CCDL. And so they're big friends of fish, as I call them, because they have really moved forward all of their software to integrate maybe different types of model systems. And zebrafish are relatively new to cancer research. So I really appreciate their dedication to being inclusive.
Mm-hmm.
I mean, it's an honor to be friends of fish.
That's oh my God. Wild. I think we posted on our Instagram recently. There's a quote that's out there, and I can't remember who it is. It might have been like Isaac Asimov that said something about, so often science is not about eureka. It's about the greatest discoveries, or when someone says, that's funny. And it feels like, who was like looking for zebrafish to share like 70% of our genome, or was it just like, what the, like, how do you stumble on that? What's your reaction to that?
Exactly. And so the field itself using zebrafish is relatively new. It was a tropical fish enthusiast that started this in the late 80s. There are two different groups that were using fish in Europe and the United States to understand like developmental processes, just really basic biology of how the cells move during when an embryo is being formed. And then really in the early 2000s is when they started being implemented to understand human cancers. And so we're really at the forefront still of implementing these models to understand, childhood cancer. And we've done a lot in terms of I study muscle cancer. There's leukemia models, neuroblastoma models, but I think there's still a lot of work to do.
Wow. Zebrafish.
That's amazing.
How does your research here fit into just the overall structure and goals of the Childhood Cancer Data Lab?
Sure. So the Childhood Cancer Data Lab has been integral really on a number of levels for me. Like Jackie mentioned, I took their, I guess was that the inaugural training?
Yes, the pilot.
The pilot training workshop. And it was really incredible. It was essentially like data boot camp.
Wow.
And so I went there for a few days, and they were trying to make us, have knowledge of how to get started on doing these analyses ourselves. And they document all of their protocols and make them publicly available. So even if you're not at the workshop, you have like step-by-step instructions on how to implement these different types of sophisticated analyses. And so that was huge. And then they're also really cool people to work with and have great ideas. And so that's important for moving your work forward. And then the other thing that they've helped me with is envisioning how the fish can contribute to pediatric cancer by looking at the transcriptome of the tumors that have generated and how closely they recapitulate the human disease. And by transcriptome, I mean the genes that are turned on and off by this human cancer gene that's in the zebrafish genome.
Wow. So what was your experience with data prior to this relationship with CCDL? Was it a big part of your work?
So typically I've collaborated. So I'm a big believer in, if I'm not an expert, I'd find the expert in order to move things forward quickly. But it's been integral to have a fundamental understanding of what needs to be done, the time required in order to do this. And it gave me a framework to build on. And now that I have these articulated protocols online, I can really go back and ask them at specific stages if I get stuck, how to move forward. And so I think it provided a good baseline for me to move forward from.
Fascinating. So where have you, so that's an example of sort of how your work integrates with CCDL. And I know it's still fairly new, but are there examples of places where you two have seen CCDL work in action successfully as you had hoped? Is there, so Genevieve, are there other examples that you've learned from, either that you hoped to or anticipated or were surprised by? And Jaclyn, where have you seen some of your most delightful successes from the system?
Well, I was so excited about taking this course that I actually sent my technician and my boss, he went to the next course. And it's kind of having a spidering effect where once you train one person, they'll train the next person. And so the impact of this is only growing. And so it's not even by taking the course directly, but by coming into contact with someone that knows how to do these types of analyses.
Yeah, so I would say it's our training effort is less than a year old, or we're coming up on 1 year since Genevieve participated in the pilot. And it's wonderful because I always like to say that no one cares about your experiment more than you. So, and I think, we spend all this time studying and nobody thinks about our project or our experiments more than we do. So to have folks who don't necessarily analyze the data themselves yet, to have them come get a little more familiar, they're pretty short, so folks don't walk out knowing how to do everything, but to give them a taste so they feel more comfortable kind of learning on their own and also more comfortable having a conversation, like Genevieve was saying, with, folks who do work on their data with them.
So participating with the CCDL, is it sort of a formal partnership or application process? Is it a tool that's accessible all of the time that somebody can log into in the middle of the night when they're feeding their zebrafish? How do you recruit new members to the Avengers?
So we do have a tool that you can access in the middle of the night when you're feeding your zebrafish, which is called refine.bio. I don't know how often the feeding of the zebrafish in the middle of the night happens.
It doesn't happen.
No, that's not real.
Brian, you have no idea.
During the day 3 times, but not at night.
Perfect.
Well, it depends on your
system. What a model system.
They're like gremlins, basically.
Right. What a model
system. Okay, so there's a tool called refine.bio, and that is the URL. So refine, REFINE.bio, BIO.
Perfect.
So that is a collection of data that's been made publicly available through researchers uploading it to government-run repositories. So what we do with that is we get that data from these government-run repositories, and then we uniformly process it. So what we mean by that is we've selected certain pipelines that hopefully have pretty general use that then act on that data. And we pull from 3 separate places. We have it kind of come under one roof, so it's discoverable all in one place. And the idea is that if you know exactly what was done to that data, you can be more comfortable and save time getting the data from us, because we're going to tell you what version of the software we used. And we're going to tell you why we did that. So the idea being, if you want to kind of quickly check something, get preliminary data for your next grant, that we've done the kind of intensive work of picking a pipeline and also the actual time it takes to process those samples, computational time. And then you can go to refined.bio, snag the experiment, and then kind of see if there's any signal, if it supports your hypothesis, if it validates something that you found in your own data set, for instance.
That's super. Brian, you taking notes?
I already went to refined.bio and typed in zebrafish. There's 638 results. I mean, my day is full.
Download them all.
So cool.
Just add them to that cart.
Awesome. What have been the most significant takeaways from CCDL so far? Do we have practical applications yet? Have there been like cool unexpected benefits or wins so far from either of your perspectives?
It's been a surprise to me, and perhaps it shouldn't have been, is the enthusiasm for these training workshops that we're putting on. People, wanting to learn this stuff. The last couple times, I believe we've been oversubscribed.
Wow.
Nice.
Yeah, so I think that surprised me, and I think I'm wrong. Like, I shouldn't have. But that was not something I was expecting going in.
Sketch this out for me. What does a training session look like? How long is it? Where is it? Are we talking about a small classroom? Are we talking about an auditorium? Are we talking about a lab? How many people? Where do they come from? How does Brian apply?
So many questions.
Et cetera.
So we're doing 4 this year. They're 3 days long. And we're doing 4 different places. So we went to Houston, which is where Genevieve's boss and technician joined us. We just got back from Chicago a few weeks ago. We're going to go to the Bay Area in September and then in Philadelphia in October. So those are 3 days long. And the way this works is the first 2 days are designed to be instruction. So we go through some of the technologies that we work on at the CCDL and are important for childhood cancer research, one of which is called RNA-seq, and now a newer technology called single-cell RNA-seq. And we show folks how to process and analyze that data and kind of give them a starting point, a set of tools. And then on the last day, that's designed for us to kind of be in the room, unstructured time, work on your own data, or otherwise practice skills that you've learned in the first 2 days.
That's what I do with my 6-year-old.
It's pretty intense. You bring your own data. And so there's practice. They have practice data that we go through together. And then on day 2, they say, go for it. And on day 3, you have to do a presentation to this group. I think it's maybe 10 to 15 people. Jackie, is that about right?
Yeah. So has been around 15 people, up to 20.
Because you said you're oversubscribed, right? Packing the place in.
It's probably more than when I was there for the pilot.
Yes, it is. It is more.
But we got a lot of attention from really smart people like Jackie. And so at the end of the day, you need to have been successful enough to have analyzed your data in order to present it to the group and talk about, it in an intelligent way, what you've done. And I would say, Jackie, you also teach good practices on how to code too, which is important.
I joke that I'm borderline militant about that kind of stuff.
Nice.
Like I need to be able to read it to find the bugs. I'm a big believer in that. It's probably a bit much.
I thought it was great.
Yeah. We can have an offline discussion about that. That's basically how I treat our workplace, and Brian really just loves it. That's really cool. So now I know Alex's funds a lot of research. Are these folks that are showing up? Do they already have relationships with Alex's? Are these mostly incoming calls? Are they outgoing calls? How are you sourcing these geniuses?
So I think, I don't have the exact breakdown, but quite a few investigators do already have some kind of relationship with Alex's. But I do believe there are some folks that, are not funded by them or are not in Alex's funded labs. But for the most part, quite a few of them have some experience with Alex's already.
Got it. And it's probably an interesting sourcing ground to find new folks as well, on the other hand, that Alex's might be a partnership with.
Yeah, it could be. I think it definitely, if you are not already familiar with our organization, coming to our training will make you familiar, for sure. Yes. We say where we're from, because that's polite and a normal thing to do when you're meeting people. So for the most part, I think people might be more inclined to either seek maybe collaborations with us at the CCDL in the future if they get to know us. Genevieve, you can stop me if I'm wrong. Maybe you're... But
no, are you kidding? I would love to collaborate.
Yeah. We're going to set it up right now on this call.
Yes.
Yeah. It's happening.
Are most of the, love the word investigators, are they US based? Are they international? What's the breakdown there?
So I think because we've been pulling, or at least contacting folks that are in kind of our sphere already, that is mostly US-based folks, because that is traditionally how Alex's has funded investigators.
Gotcha. So let's talk about the hard stuff. Where are you both? And I realize, again, you don't do the same thing, but your jobs do complement each other, both theoretically and practically. Where are you running into obstacles? Do you find things running into the ground or have been difficult so far with CCDL or Genevieve with your own research, either in collaboration with CCDL or otherwise?
That's a great question.
Yeah. Go ahead. I'm sorry.
Go for it, Jackie.
So I would say what I didn't talk about, something that also surprised me going back to a little bit what we were talking about earlier. One of the projects that I finished up and published in the last year was a project that essentially looked at training a machine learning model and extracting patterns from a large collection of essentially random human data and then applying it to rare diseases like childhood cancer. And what we saw is essentially that you were able to learn kind of more pathways or get a higher resolution look into the rare disease data, even though that model had never seen the rare disease data before. So that's been really exciting, and I think speaks to how something like Refine.bio that we're using could be really useful, even though the bulk of the data that's in that resource is not specifically looking at childhood cancer data. So bringing kind of this new idea, this conceptual advance, if you will, that was a challenging project and really exciting project to work on. And yeah, I'm excited to do that, maybe looking at zebrafish in a similar way and kind of transferring over what we learned to human tumors. But I think all of that is very new. And sometimes when things are new, they're hard. But definitely an exciting place to be.
I think one of the biggest obstacles that we face is really understanding what genes are doing in a tumor. And so when you go to a clinic and you get clinical genomics done on your tumor, you get a report with different mutations in cancer-causing genes. But if these mutations are one base over and they don't have a described animal model, then you can't call them as being causative. So
could you explain that for a basic human being?
Sure. So a lot of times we get these reports back from clinical sequencing efforts, and you just have gene lists, and there's a bin of genes that you found in exact mutations in other tumors, and these are causing the tumor probably. But then you have a bunch of passengers, and which is mutations that we have no idea what they're doing, but they're there. And so that's really why I've tried to integrate myself into a clinical community, because what I can do with the fish model is take these mutations that we're not sure what we're doing and then build zebrafish avatars of human disease and try and understand how the genomics of a human tumor will play out in an animal model.
Interesting.
Wow. And so it's a way to functionally validate what we're seeing from patient tumors, putting it into the fish, seeing the presentation of the disease, potentially how it responds to different therapies or combinations of therapies. And then, the goal for me would be one day moving that back into the patient after testing it in the fish.
How often does research or results not translate from the zebrafish over to humans?
Well, it happens in all models, right? So for example, the sepsis model in mouse wasn't predictive of what was happening in humans. Mm-hmm. And so it's bound to happen in the zebrafish as well. There was a really exciting paper recently where they took a specific mutation they found in a human with a lymphedema disorder, put it into the fish, screened drugs on the fish, found a drug, and put that drug back into the single patient and improved their course of disease. And so it is possible to go from fish to human directly in terms of drugs that you're finding that are potential therapeutic options. And so for drug development in cancer, there's a lot of failure once you reach clinical trials. And so to me, the best drug has activity in multiple model systems, and that's the burden of proof you should use to move forward.
Sure. I was just thinking, someone created some frustrated researcher who I'll have to find this, created basically a Twitter bot.
In mice.
And I just thought it's so great because we talk about that all the time, and it's easy as I go through and try to find the biggest news and most impactful news, but also the most truthful and validated news, and often we're linking straight to journals and things like that, because you see these headlines sometimes in respective publications, because they'll go from, a paper to a journal to getting picked up by a science blog to getting picked up to the New York Times. And then it's a headline like Alzheimer's cured. And you're like, what the fuck? But if you really dig into it, it's like Alzheimer's cured in mice. And, and I just, again, the context is helpful. And that's why I was just curious. Because again, we use mice because it is so close and it's really crazy. And the zebrafish, obviously is proving similar, but obviously there are going to be times where it doesn't translate.
Absolutely. And so that's why it's really important for me to validate everything I find in patient tumor data. And so are these genes important for overall survival in patients? And that's my stopgap essentially to, do I move forward with this or not? And so that's why I've really partnered with the CCDL to filter the things that might be most important to move forward with, because I don't care about fish disease. I care, right?
Look, fish are
I do care because I want them to be healthy.
Sure.
I want them to get cancers that recapitulate human disease.
Brian, how many zebrafish have you put in your Amazon cart since this conversation started?
Is that how you buy fish? Come on.
You just go to your local pet store. It's the same fish. It's like an inch long.
I saw it. It's beautiful with its little stripes. I grew up with a lot of fish and fish tanks in my house, and I loved it. You could just sit there and stare at them. They're beautiful.
You could have been solving cancer this whole time.
I was like 11.
I don't want to hear any excuses. Do you think these ladies made excuses for being 11?
No, I'm sorry.
Well, you know what? We'll take this off. We'll deal with this ourselves.
I'm sorry, doctors and Quinn. Do you know? Has CCDL inspired other organizations to undertake similar endeavors? Or if not, where could you imagine this model going into other areas of cancer research or just medical research in general?
It's a really great question.
Thank you.
I don't know. Yeah, I'm not sure that I've seen this exact model. It's still early days. But I think it is useful to have an interdisciplinary team that, has funding from a research organization to do this kind of work. I could see, of course I'm biased, but I could see this model being really powerful because there are some problems that are difficult, like require a lot of resources, and that they won't necessarily get attention in, academic institutions or industry or what already exists. So I could see it being something that crops up more. I think hopefully, we're very successful and people look at us and they're like, oh, we want to do that for our community. So that would be great. But I do think it's early days. I think early days or not, we've processed a lot of samples. We're getting our training program up and running, and I'm super excited about it. So I hope we are in a place where folks look at us and go, wow, I really want to do that for our community.
I don't know if you guys have this in Philadelphia, but Brian is quite good at, do you guys have the guys who stand in the street corners that like flip the arrow sign up and down and dance with it to point people to come to their car wash? He could do that for your training sessions. But if you guys don't have those, I can think of another analogy maybe that's appropriate. Right. He's like, you talk about interdisciplinary. He's got a lot of skills.
So I think we're a little tight on sidewalk space.
Sure.
Got that move.
Okay.
But like, get him a clipboard maybe. Okay. The clipboard model is quite popular.
So typically CCDL has information booths at big cancer meetings. So I see a lot of opportunity there for sign flipping.
Sure.
That's true. We should fly you in.
I mean, I'm in. I was kidding 10 seconds ago. Yeah. Now I'm not. This is dead fucking serious. We will come and wear t-shirts or crop tops or, whatever gets the job done.
Well, we should do some user testing and see what really works.
Let us be your
We're about that here.
Jay also has to be involved in this. So I'll make sure to rope him in without telling him.
Good. Yeah.
Good plan.
Great plan.
All right. So let's get into our action steps. We like to provide very specific action steps, if we can, that our listeners can take to support your mission with their voice, their vote, and their dollar. So let's get into that. Let's start with voice. What are the big actionable specific questions that we can all be asking of our representatives that would help support your missions, ladies?
Literally, what should they say?
Okay, so obviously there's federal funding agencies for scientific research, primarily the NIH for health and the NSF, and advocating for increasing the budget for both of those organizations makes a really big impact on what we're able to do. Right now at the National Cancer Institute, which allots grants to researchers at academic institutions, the funding line is, I think, at 8% of submitted grants are accepted for funding.
Jeez.
And so we're missing out, and it's an even smaller percentage that's allotted to childhood cancer, but we're missing out on a lot of great science because of that. And it's a budgetary constraint.
I was going to say, so yeah, is it just purely a funding constraint that knocks out 92% of requests? Is it a low quality of entry or?
No, there's a lot of great science that is being missed out on, and it's just the budget is what it is, and so it's getting more and more competitive now to get funding, and that's why, the foundations like Alex's Lemonade Stand have made a huge impact on what I'm able to do, and so the grants that they give for young investigators really propel them to be able to do the kind of creative translational research. And then they have an A award that propels you from a postdoctoral position to faculty as assistant professor. And so they're really trying to build the next generation of cancer researchers.
I wish there was a way to almost make some publicly available / searchable list. Almost like, I hate to use this as an example because it can go wrong so easily, but a Reddit almost upvoting system of like all the projects and research that don't get funded. That, like you said, so much good science is not happening. To put those out there so people see, like, hey, this is why you need to get your representatives to vote for more funding, because these are quite literally these specific things that are not happening because of that.
Exactly.
Brian, can you put that on your list?
Absolutely. And I want to go to dollar too. What can everybody do to support you with our money?
If only there was like a foundation that you guys haven't mentioned where people could send their money to.
I have a feeling that there is, so if we could just talk about it for once.
I don't know.
Sure. So, Jackie, do you want to take this?
Yeah. So,
bam.
Alex's Lemonade Stand Foundation. Bam.
Bam. What's the URL for Alex's Lemonade Stand Foundation?
alexslemonade.org.
Beautiful. This is the moment where I'm going to pimp LA Loves Alex's Lemonade Stand Foundation. Have either of you come to that event yet?
Incredible.
Yes, it's amazing.
Yes, it was great.
Are you coming this year?
I don't think so, and I'm disappointed.
I am not. Yeah.
Whoa.
Wait,
they're busy.
Why not? What else you got going on? Yeah, I know, but it's a fun time.
It is.
I would love to.
It is fantastic. It is one of our favorite things. Brian, when did I indoctrinate you into this? Yeah. Last year or 2 years ago? It is such an impactful, moving event. For everyone, it is, what is it, Brian? September 14th this year?
Yep.
Is that right? September 14th, Los Angeles. It's at UCLA. Regular tickets are like $100 or something like that. You get unlimited food handed to you by like 100 of the greatest chefs in America / the world. There's like 20 incredible mixologists. There's a wine area, and then there's also Jay and Liz and their children, their sons up there talking, and other very small cancer patients and survivors up there talking, and you're drunk and crying and just throwing money at this incredible organization. And it's sunny and it's beautiful, and everyone is having just the greatest time for such an incredible event. And I can't recommend it enough. So if you've never come, please check it out. It's called LA Loves Alex's Lemonade Stand. The past few years, they've raised $1 million on the day, each day, for efforts like this to fund CCDL and other research. And they also support travel funds for families that can't afford it, because it is an unexpected life expense, and it's really fucking expensive, and the whole family's affected. And someone's got to help out with that, and they do. So, yeah, please check it out. It's pretty fantastic.
We'll be there. Important, not important.
We will definitely be there. Yeah, and we will. If you come, we will find you. Not in like a not like a bad way, but like an exciting. We'll take a picture together way.
It sounded ominous, but
no.
We just want to eat food with you and drink drinks with you and cry with you. That's all.
And thank you for what you're doing. Yeah, a lot of crying. Brian, take us home.
Ladies, seriously, thank you so much for being here. This has been so fantastic. And I can't wait to see what else comes out of the CCDL as it grows. Can't believe it's only, did you say a year or not even a year yet?
August 1st, 2017, I believe.
Wow.
Yeah.
What year is it? I don't even. It's God!
It's almost
question. 2 years old. We're gonna have a birthday.
Almost 2 years old. Yeah.
We're gonna have a birthday party.
There will be a time when we email you after this conversation to say, hey, who else should we talk to? Do you know anybody else that is trying to change the world and solve kids' cancer or cure
anything?
I mean, anything. Just all of the things.
Yeah, people who inspire you.
If there are people that come to mind, please pass them along. It's how we get some of our absolute best guests on the show. And now it's time for our Not Lightning Round.
Not a lightning round. It's just a couple more emotional questions.
Yeah.
And then we'll let you guys get out of here. To both of you, to each of you, when was the first time in your life when you realized you had the power of change or the power to do something meaningful?
Yeah, so I think it went back to, what year was the Young Investigator Summit in LA, Genevieve? Do you remember?
Oh, gosh. I think it was 16 or 17.
17? Yeah, just like meeting all of these investigators who were about the same career stage as me, because I was a postdoc at the University of Pennsylvania at the time. And just meeting all these childhood cancer researchers and like their passion for their research was just like infectious. And like understanding that this initiative was getting underway and being part of this exciting science, helping these folks like do what they're so passionate about and what really answer really important questions. That YI Summit, and that's where I met Genevieve, for the record, too, was really wonderful. I think it was such a great introduction to the foundation, and is why I'm here, just working with these scientists who are so talented, so passionate. And I was like, oh, I can help them. I can be part of this. So
awesome.
I'm trying to think of a specific instance. Like I said, my biology teacher was really influential on me in high school, and I was always gravitated towards incredibly challenging problems like childhood cancer, because I feel like I can make a big impact in the area. And I think this manifested early, because this is really random, but in our senior year in high school, we had to give each other awards. And this person I barely knew gave me an award that she had self-named, that I was the most determined person. And
wow.
And so I think it's true. I'm very determined. And so I think I just have been manifesting that for a while, unintentionally, but but I am very focused. And, I do think I can make a big impact by staying the course.
That's awesome. Since we're talking about it, my senior awards were most hair color changes and craziest.
Wow.
Oh, thank you, Brian. That's so helpful.
Well, we were on the topic.
Yeah. People whose name starts with doctor. Let's limit this one to that.
Got it.
Yep. Who is someone in your life that's positively impacted your work in the past 6 months?
Oh, that's easy for me. My postdoctoral mentor is James Amatruda, and I highly recommend you speak with him as well. And so he's the most intelligent, kind person that you'll ever meet. And he's been just completely instrumental in letting me have freedom of thought and be independent. And now I'll be starting my own lab in the next 6 months. So he's an incredibly inspiring person.
Awesome.
You should absolutely talk to him. He's great. It was so rad having him for training.
Oh, cool.
Yeah. So I would say the director of the CCDL is Dr. Casey Green. And I was a postdoc of his before I got started here at the CCDL full time. And yeah, his vision for the lab and kind of shaping it and kind of assembling this team of designers and developers and scientists. It's been incredible to watch and to be a part of.
I love it.
Awesome. Ladies, what do you do when you feel overwhelmed? When it's all too much and you need to chill and relax?
Sure. So I recently joined this women's only gym, and I do like fitness classes there. I had one this morning actually. And so that's a good way, to burn off some energy, really.
Oh, yeah.
What's your class of choice? Is it like Zumba, Pilates? Like, what are we talking about?
No, it's called, it's actually designed for athletes. We do like rowing, run wind sprints, a lot of lifting. And so, I do the whole spectrum. And
cool.
So it's been fun. You have to challenge different parts of your brain. And so the other thing I do is take naps sometimes.
Yeah.
I love a nap.
Nice
cool.
Nap. I love to go outside and hike too.
I love that. I've been trying to get Brian to do, what's the word, Brian? Cardio?
No, get out of here.
It's new to him, but we're working on it.
He tries to get me to do these Spartan races where you're, running for days and like you're in mud and you're climbing walls. Let me run on the treadmill.
Okay. Baby
this
steps.
Is consistent with Jay's kind of comments about Quinn and
see?
That's unfortunate. I did not know with it. He told you. I've got some fun plans for him for this year. It's going to be great.
Great.
I won't tell.
Yeah, please.
I think my answer to that question proves why Genevieve was most determined and I was not as a senior in high school. But yeah, I have been very much enjoying Red Dead Redemption 2.
Oh, it's so
right.
Yeah. That's been really, past couple months. It's been great to kind of escape to that.
Can you do that in VR yet? Is that a thing?
I have no idea.
I just want to be in that world.
It's beautiful. You
Wow.
ride a horse.
Yeah.
You know.
So my buddy, friend of the pod, Seth, his one of his good friends voices the main character, and
oh my goodness,
and has said he basically can't go out in public because the game. I mean, you know, so many people have played that game, and not just played the game. We're not talking about Mario Brothers. They've spent hundreds of hours of their life with this game.
So many hours.
And so the second he opens his mouth, people are like, "Holy shit!" Like blows movie stars out of the water.
I feel so ignorant because I did just have to Google what that was. So I'm a little embarrassed by that.
No, it's okay.
It's okay.
It's okay. It's okay. It's pretty fantastic though. But yeah, I was just like, God, that's such a weird thing. Anyways, great game.
Wow.
Such a good game.
Okay, Brian.
Great game. Here we go. If you could each answer this question for me, please. If you could Amazon Prime one book to current president of America, Donald Trump, what would that book be?
Mm-hmm. We've had everything.
Nonviolent communication is what popped into my head.
Excellent. I was going to go with Alex and the Amazing Lemonade Stand, just to show how one person can make a positive change on this world.
That's awesome. Both of those are fantastic. We've had everything from the Constitution to coloring books, just because.
Everything.
Awesome. Well, listen, this has been long enough. There's all kinds of cancer that hasn't been cured since we've taken an hour plus of your time. So we're going to let you guys get back to it. But we cannot thank you enough for your time today and for everything you're doing with each other in your own endeavors and what's to come, because I know you're just getting started. No pressure. So thank you. And I hope to see you at an Alex's event this year, next year, somewhere. And when you get that lab started, put in a couch because Brian will come sleep on it.
And I'll do the sign spinning thing.
We'll do the sign
definitely.
spinning.
You're welcome. Sure. You're welcome to come join.
It'll be great. He'll do any job. Thank you guys so much. We really appreciate it. And we'll check in down the line and see how everything's going.
Thank you.
Thank you. Thanks for having us.
Thanks to our incredible guest today, and thanks to all of you for tuning in. We hope this episode has made your commute or awesome workout or dishwashing or fucking dog walking late at night that much more pleasant. As a reminder, please subscribe to our free email newsletter at importantnotimportant.com. It is all the news most vital to our survival as a species.
And you can follow us all over the internet. You can find us on Twitter at importantnotimp.
Just
so weird. Also on Facebook and Instagram at Important Not Important. Pinterest and Tumblr, the same thing. So check us out, follow us, share us, like us. You know the deal. And please subscribe to our show wherever you listen to things like this. And if you're really fucking awesome, rate us on Apple Podcasts. Keep the lights on. Thanks.
Please.
And you can find the show notes from today right in your little podcast player and at our website, importantnotimportant.com.
Thanks to the very awesome Tim Blaine for our jammin' music, to all of you for listening, and finally, most importantly, to our moms for making us. Have a great day.
Thanks, guys.